Why is cellular aging important? Cellular senescence, while complex, plays a key role in aging and disease. Originating back to 1961, this field has advanced beyond research labs and is now a key focus in healthcare and biotechnology. Cellular senescence is when cells stop dividing, a permanent yet reactive biological response to stress. These stresses can be natural, like telomere dysfunction, or external, such as DNA damage or the activation of cancer-related genes. Initially, cellular senescence was seen as a main cause behind tissue aging. However, this perspective has shifted, recognizing a more complex role of cellular senescence. It serves both as a protector and a contributor to age-related decline. Cellular senescence shields the body by stopping the division of damaged cells, preventing cancer. However, with time, these non-dividing cells pile up and hamper tissue function and regeneration, contributing to the slow decline seen in aging. The paradoxical role of cellular senescence aligns it as a primary focus of modern science. As we delve deeper into the world of cellular senescence, we embark on a journey that intertwines biology, medicine, and technology. It's a journey that promises to unravel some of the most enduring mysteries of human health and longevity. The journey to cellular senescence begins with an array of triggers. These include DNA damage. This is a primary catalyst for senescence. Cells have intricate mechanisms to repair DNA, but when damage is severe or repair mechanisms fail, the cell shifts to a senescent state to prevent the propagation of damaged genetic material. Telomere dysfunction. Telomeres, the protective caps at the ends of chromosomes, shorten with each cell division. Once they reach a critically short length, they signal the cell to enter senescence, thereby acting as a natural limit to cell division. Oncogene activation. Certain genes, when mutated or overexpressed, can drive uncontrolled cell proliferation, a hallmark of cancer. To counter this, cells may enter senescence, halting potential cancerous growth. Stress from dysfunctional organelles. Cellular components like mitochondria, when dysfunctional, can lead to oxidative stress and further push cells into senescence. One of the most significant aspects of cellular senescence is the senescence-associated secretory phenotype, SASP. Senescent cells secrete a cocktail of inflammatory cytokines, growth factors, and proteases. While initially part of a protective response, the SASP can, over time, have detrimental effects. It can create a pro-inflammatory environment, contributing to the development of age-related diseases. The SASP factors can alter the tissue microenvironment, impacting neighboring cells and sometimes even promoting tumor growth. In a paradoxical twist, while senescent cells themselves are resistant to cancerous transformation, their secretions can influence the behavior of nearby cells, potentially leading to a pro-cancer environment. Understanding cellular senescence is not just about looking at cells under a microscope. It's about piecing together a puzzle that spans from the smallest units of life to the entirety of the human organism. The dual role of cellular senescence, from protection to aging. Cellular senescence, originally evolved as a protective mechanism against cancer, has a paradoxical role in the human body. Its dual nature unfolds in the way it protects tissues and contributes to aging and disease. The primary evolutionary advantage of cellular senescence is its role in tumor suppression. When cells incur damage that could lead to malignant transformation, such as DNA damage or activation of oncogenes, they enter a state of irreversible growth arrest, senescence. This mechanism acts as a critical barrier against cancer, preventing damaged or potentially cancerous cells from dividing and proliferating. Besides cancer prevention, senescent cells play a significant role in wound healing and tissue repair. By secreting various growth factors and cytokines, these cells help in coordinating the tissue repair process. For instance, they can recruit immune cells to injury sites, stimulate new blood vessel formation, and aid in the remodeling of tissues. In a fascinating twist, senescence is not just a feature of aging cells, but also plays a part in embryonic development. Senescent cells have been found in developing embryos, where they influence tissue development and organ formation. This involvement underscores the complexity and nuanced roles of senescence beyond its traditional association with aging. Senescent cells, through their SASP, release a mix of inflammatory cytokines, chemokines, and proteases that progressively damage tissues and disrupt normal cellular functions. This chronic, low-grade inflammation is a significant factor in many age-associated diseases, including cardiovascular diseases. Senescent cells contribute to the hardening of arteries and the development of atherosclerotic plaques, increasing the risk of heart attacks and strokes. Neurodegenerative disorders, inflammatory factors released by senescent cells can exacerbate the decline in cognitive functions, contributing to diseases like Alzheimer's and Parkinson's. Osteoarthritis, the degradation of joint tissues, often seen in osteoarthritis, is accelerated by the inflammatory milieu created by senescent cells. Therapeutic approaches, senolytics and xenomorphics. Senolytics are drugs designed to selectively induce death in senescent cells, thereby reducing their burden in tissues. This approach is based on the unique properties of senescent cells, such as their resistance to apoptosis, programmed cell death, and their distinct metabolic pathways. Drug development and trials. A variety of compounds, including dasatinib, quercetin, and fisetin, have been identified as potential senolytics. These compounds target various pathways to induce apoptosis in senescent cells. Clinical trials are underway to evaluate their efficacy in treating age-related diseases and improving overall health in the elderly. Mechanisms of action. Senolytics generally target survival pathways that are upregulated in senescent cells. For example, some drugs inhibit the BCL2 family of proteins, which senescent cells often rely on to evade apoptosis. By disrupting these pathways, senolytics can trigger the death of senescent cells, potentially alleviating their negative effects on aging and health. The development of senolytics and xenomorphics represents a groundbreaking shift in our approach to treating aging and its related diseases. By targeting the cellular underpinnings of aging, these therapies offer the potential for a paradigm shift in healthcare for the elderly. The field of cellular senescence is rapidly evolving, with promising avenues for future research and therapeutic development. As we deepen our understanding of the nuances of senescence, the potential for impactful medical breakthroughs increases. Thanks for watching. Remember to like, subscribe, and comment.